A very good day to Dr. Arnold and Dr. Wanfami. My name is Michelle Samkavan. Today, I'm going to present about DNA ligation. So in my presentations, I will include about the overview of DNA ligations, the molecular basis of DNA ligations, the two emergence of DNA ligations, research about DNA ligation, and also my conclusions. And I will also attach my reference in my presentations. So now, let us go to the first part, which is overview of DNA ligations. So generally, the first illustrated person is Arab Lemon, which is the guy in the photo. The principle of DNA ligation is to join two DNA fragments through the formation of phosphodiester bonds. So in this process, DNA ligase act as very important role to help in joining two strands of DNA fragment which covalently join the phosphate backbone of DNA with either blood ends or sticky end. In another word, we can call sticky end is cohesive end. So there's a lot of application of DNA ligations used in this century, such as cloning process, recombinant plasmid, and a lot more. Before we go to the process of DNA ligations, let me talk about some principles of DNA ligation that we have to understand. So the first one is blood ends. Blood end is when both of the DNA strands are cut at the same positions and there is no nucleotide are left unpaired at the end of the DNA strand. Well, sticky end happen when each strand of the DNA are cut in the different position. So, one strand are overhang by the several nucleotide. This single strand can be spontaneously paired with each other. So this is what happened in the sticky end. So now we will go to the process of DNA ligations. So the first step of DNA ligation, it will happen enzyme annihilation. Enzyme annihilation is when the nucleophilic attack to the alpha phosphorus of ATP by ligase and result the release of the pyrophosphate group, which is PPI, and the formations of the covalent ligase annihilate intermediate in which AMP is linked via a PN bond of a lysine. So what happened in the second step? Second step is happen where aniline will transfer to the DNA, where AMP is transferred to the 5' end of the 5' phosphate terminal DNA strand to form DNA anilate. In these reactions, the 5' phosphate oxygen of DNA strand will attach to the phosphorus of ligase anilate and the active site lysine is relieved from the group. So the third step, which is the last step too, it happened Nick sealing. Nick sealing is when the ligase catalyzed and attacked by the 3' OH of the nick of the DNA anilate to join the polynucleotide, and the AMP will release. So now I will briefly talk about the factor affecting ligations. So the first factor is DNA concentration. So when the concentration of DNA is high, the chance of one end of DNA molecule meet the end of another molecule is lower. Therefore, it will form an intramolecular ligation. In other words, when the concentration of DNA is low, it will form intramolecular ligations. So the second factor is ligase concentration. The ligase concentration is directly proportional to the rate of ligation, where when the concentration of ligation is high, the rate of ligation will also be high. The third factor is the period of incubation. The period of incubation will affect by the iodine strength of a buffer. The last but not least is the temperature. Temperature is depends on the number of hydrogen bonds in the ligations. So it will increase the melting temperature of ligations when the number of hydrogen bonds is high. So now we have go to the part 3 of the presentation, which is tool and machine of DNA ligations. So by using a conventional technique to handling DNA ligation, to conduct the DNA ligation, the material that we need are T4 DNA ligase, buffer vector DNA, insert DNA, nucleus-free water, T4 DNA ligase. So these are the steps that needed to handling the DNA ligase. So the first step, we mix all of the material by pipetting up and down in microfiche briefly. Then the second step, before we go to the second step, we have to know the resection site. We have to know the type of the resection site that cut by the resection enzyme is either blood end or sticky ends. Both of the ends can be incubated at 16 degrees Celsius overnight, or in another way, in alternative way, we can incubate the bud end in the room temperature for 2 hours, or sticky end we can incubate at the room temperature for 10 minutes. So the third step is the heat activate 65 degrees Celsius for 10 minutes. So basically DNA ligation can be done within these 3 steps, and the fourth step we can proceed to our transformations. So nowadays scientists had explored more and more convenience kits that help the DNA ligation process can be done more fastly, more rapidly, more conveniently. So one of it is rapid DNA ligation kits, where the DNA ligation can be done in only 5 minutes at room temperature, and inside the kits contain T4 DNA ligase and a special formulator 5 times rapid ligation buffer optimized for fast and efficient DNA ligation, and it can directly use for bacterial transformation. As a reminder, the lower temperature will stabilize the hydrogen bonding between the complementary nucleotide, and this can ensure the quality of the ligation. But as the temperature is lower, the time needed for incubation is longer. Okay, now we have reached our next part of our presentation. So in this part, I will give an example of the research about DNA ligation. The title that I choose is DNA ligation using a disposal microfluidic device combined with a micromixer and microchannel reactor. So in this research, the scientist is to examine DNA ligation using the device, and these are the material that used in this research to conduct the experiment, and they also include the methods, the result, the discussions of the experiment, and finally they have concluded that DNA ligation was successfully accomplished in the integral microfluidic device, with the time shortened from 4 hours to only 5 minutes. So this is not only are able to shorten the time, this is also will increase the efficiency of DNA ligations. At the same time, will will ensure the quality of the DNA ligations. So now we have reached our last part of our presentation, which is conclusions. So as the technologies goes, there's a lot new technique of DNA ligation has been discovered with more convenient and faster. So it is very important that we know how to choose a right method for our study to ensure the quality of DNA ligations. Besides of DNA recombinant replication and cloning, DNA ligation also help in DNA repair, DNA damage, and DNA nick. So if there is no DNA ligation, what will happen? It will lead to disrupt of gene expression and will cause the genetic disorders such as Lig4 syndrome. So it is very important. So this is the last part that I will attach my reference that I used in these presentations. So this is all from me. Thank you for your attention.